What is the industry standard of brain implants? Meet the Utah array, an array of 100 silicon electrodes implanted in specific regions of the brain. It is clearly the gold standard of brain-machine interfaces, but stiff electrodes trigger foreign body response and glial scarring, which is when the immune system covers the implant, isolating it from the rest of the body and making it unusable. Could we build a Utah array but with living cells that integrate into the tissue? Absolutely, and it has already been done at the University of Utah and TU Dresden. What they've built is Biohybrid Transition Microelectrode Array, or also called TMEA. The device has two parts. First, the base, a silicon chip with a 4x4 array of recording sites, and each with 25 gold-coated microwells, 400 in total, and each 30 micrometers in size, custom made to hold single neurons. And second, the shacks, which are polymer needles, 3D printed, and they have 40 micrometers at the tip, 1 millimeter in length, and they are empty inside. So the system works like this. They have prenatal mouse cortical neurons at the top, and the neurons project their axons through a hydrogel channel inside the needles, inside the brain. And the question is, does it actually work? We need to answer three questions. Can it be inserted without breaking? They simulated and physically tested the shanks using a pneumatic insertion technique on a rat cadaver, and all shanks were intact upon piercing the brain tissue. No buckling. Second, how long does it last? They soaked the devices, without neurons or hydrogel, in a hot salty solution for 46 days, which simulates one whole year inside a human body. And this is called an accelerated aging test. The impedance dropped just slightly from 16.5 to 15.5 kiloohms, and the charge storage capacity was stable, and the device was viable after this for one whole year. And third, are the neurons alive? To know that, they stained them and used fluorescent confocal imaging. They loaded the neurons inside the polymer shanks with hydrogel, and they saw that after 1 week the axons projected outside the shanks to a brain phantom. So they saw that they could detect neural signals from the base. That's what they tested. So next, it's going to run preclinical trials and measure and try to evaluate further the biocompatibility. And succeeding at this would mean much better temporal resolution than conventional approaches, because you potentially increase the amount of readout points in a specific area. And they are also considering making the shanks biodegradable, leaving only the neurons reaching the brain and the base. And that's a really cool paper. And I can't wait to see more progress coming from these people. Anyway, subscribe if you want more. Thanks for being there. Cheers.